Okay, so the first of these lectures about cellular review, we just went over some of the major components, just reminding you about the forms of DNA, what DNA looked like, what chromosomes look like, and where they occurred. I thought I would just give you a very few examples of some cellular pathways to kind of elevate our thought process about it a little bit, and thinking about how what genes do inside of a cell, right? Obviously, genes are making proteins, one of the things that they do, and then proteins are part of these big pathways. So let's just look at a few of those. One of them is protein fate, autophagy, and autophagy is basically, think about it, we make all these proteins, right? We go through the process of transcription, translation, make these amino acids. Sometimes we do a great job. Sometimes we don't do a great job. Sometimes they're misfolding. Sometimes we produce a lot and then the environment changes and we don't need anymore. So even though they were produced correctly, we don't need it. Some just are used a few times and then age and no longer can do their function and we need to get rid of them. So what do? Well, we can't just pile all that stuff in the cytoplasm. It has to be removed. And the way it's removed was by the process of autophagy relation movement. And so what we're going to produce is an autophagosome. And the autophagosome is basically a membrane, a vesicle membrane that collects the materials that you're trying to get rid of. They collect in this vesicle. Vesicle is then moved close to the nuclear region. In that process, a lysosome is brought in and combines with it to perform what's known as an autolysosome. And that lysosome is what it says, it lyses, right? So it comes in and breaks apart the amino acid chains or whatever else the organelles that we're trying to work with, and releases those, allows those to be released so that they can be reused. So it's a recycling system that's found, okay? Now the pathway looks something like this. The pathway for autophagic degradation looks about like this. Holy cow, okay? Why am I showing you this? Well, I most certainly don't expect you to memorize this stuff, okay? There's no reason for that. I'm trying to show you the proteins that are necessary and the relationship back to genetic control and how all this works, right? Well, I mean, we've always thought of, simple genetics, okay? Now you've already dealt with pathways, right? You had to learn the Krebs cycle and you've learned some other cycling type pathways where we get hydrogens and things thrown off and all that, energy pathways. But biological systems have these huge signaling pathways they're really complicated. So even though you may have had biochemistry, I don't know if you've put some together that look like this. Again, I'm not expecting you to memorize this. I think you should note that it is a pathway for degrading proteins and other things. But and there's a lot of signaling that goes on. Okay, you can have insulin signaling, you can have hypoxia, you can have lack of nutrient availability, energy. ER stress, so the endoplasmic reticulum can be stressed. ROS, reactive oxidative species that are produced, mitochondria. One of the things that happens when in energy production in the mitochondria is that you get ROS produced, right? And you get ROS from other processes. If too much of that comes along, it forces the system to respond. The system usually responds through a MAP or MAP kinase, or PK alpha kinase system, and you get these triggerings of these big pathways. That's what all these things are up here, these complexes. And depending on what the response is, what these are required. That's what it's showing off over here. So pretty much all of this part of the pathway is involved in endoplasmic reticulum and taking those membranes and starting to pull them out and form them so that they can start to form around the material that you're trying to ultimately make into a lysosome. And then there's other parts of the pathway once you move down through here, where you, in the cytosol again, where you start to move it. And finally, you'll start producing the autophagosomes, and then the lysosomes will come in from other areas. There are genes involved in that. You get a fusion. Once you get that fusion, you start to break down, okay? Now, so what, okay? Well, so what is that? Each one of these green little boxes is a protein. Right? Now, some of them are units where you've had a single gene, and then you just had alternate splicing of combinations. Some of them are unique. Many of them have unique genes. So just in this one pathway, just cleaning up these structures requires all of these genes to be activated at the appropriate time and form. And so imagine you can get lots of different kinds of genetic problems if you simply fail. You just don't get the right cleaning up, and that's one of the things that we'll see in syndromes. Somewhere in this process, we'll see a failure of one or more proteins to be produced. You won't get the proper formation of the autolysosome or something else. You'll get an ultra buildup of these proteins that you don't want, or variants, defective parts of proteins, and you'll end up with certain types of genetic diseases. And it can be because many of these genes can be mutated, lost, modified, or simply not function properly. Now, one of the cool things here is, of course, is that how does this all happen, right? How do you get, once you get this stuff, how does it move around? Well, it moves on these microtubules. Now, we've already talked about microtubules being important, a version of them being important in the process of mitosis, right? And meiosis, of pulling apart the sister chromatids or the tetrads. But the way they move is by dynein. And dynein is a protein that group that tends to collect objects that you want. Can be it could be a lysosome, can be the autophagosome itself, can be just the protein itself, or it can actually be mitochondria. And it sort of walks its way down the microtubules, okay, electrostatically, and you've probably already seen this somewhere. Kinesin tends to walk away from the nucleus to the outside, so it transports things toward the cellular membrane. Microtubule then, of course, run both ways. They can be stretched each way. Dynein is responsible for moving it toward the nucleus, and where areas, particularly things like where you're going to have lysozyme activity. And dynein is pretty cool. If you get a chance, you just put in dynein or something into YouTube, and you can actually see a little model of how it walks down through there, mechanically walking. Okay? Now, one of the things that, from a genetic sense, that makes this work is that obviously when you connect up dynein here to the whatever it's trying to pull around with it, it doesn't do it by just sticking to it. It's not Velcro, right? But you need something, you need an adapter protein to make it work. And one of the common adapter proteins is HDAC6. HDAC is a histone deacetylator. Okay, that's we've talked about, and we will talk more about histone deacetylation. It's one of the ways you open up DNA, right, or close DNA down. And HDAC families are that. This HDAC6 is unique in, well, it's one of the unique versions, because it only stays in the cytoplasm. So once it's produced, once it goes through the process of translation, it has what's known as a nuclear exclusion region, a domain. It can't get into the nucleus. So unlike the other HDACs, which are produced in the cytoplasm and moved in to do their job as far as the deacetylating or whatever they're going to do, it can't move in. The gene itself, HDAC6, is found on the X chromosome. It's found up on the short arm. That's where a little arrow is here. This is centromere. That's found up about, I think, 1128, 29, somewhere like that. So region right up in this region right here. And it's a pretty cool protein. It has an adapter region, ubiquitin binding region. And what happens is that when proteins become disformed, okay, they misfolded or whatever, there's a system that will ubiquitinate them. Okay, we'll add ubiquitin chains to them. HDAC is able to recognize through this binding domain those ubiquitinated regions, and that's what it attaches to. And then it brings and helps transport that material to the dynein structure, okay, and helps act as a intermediate, okay, so a linker protein, and it forms up with the dynein. The dynein then can move it down, and these ubiquitinated structures become recognized as part of the lysosome or autophagic system. Okay, it will not only do this for proteins, it'll also do it for mitochondria. So we see, I told you, that we had the P62 involved, but HDAC6 actually turns out to also, once you get rid of depolarized mitochondria, it will actually attach to it. Anyway, it's an interesting gene. If we look at the domains that are found on it, so you take the gene and actually look at what the protein's productions are. It's got a cytoplasmic anchoring domain, but it's got two areas of catalytic activity. So it's an enzyme. At least parts of it are functionally as an enzyme. One of them interacts with alpha tubulin, so helps get this tubulin formed here, and the others do some other activities. It's ubiquitous, found throughout the body, and almost all tissue types have HDAC6. In fact, all do that I know of. And mutations of this gene, okay, where you mess up the domains or something like that, are associated with neurological diseases, because remember, it's part of transport. So a number of neurological diseases can be associated with malfunctioning HDAC6. If it's not adapting properly, you can get too much buildup of protein aggregates. Neural cells in particular, they don't just let them build up in the cell and the cell get bigger and bigger. They tend to get rid of them. They stick them on the outside of the cell. And some places that's okay, but in a neural cell where you're trying to fire between one neuron structure and another, if you got a bunch of protein piled up around one, it's trying to fire through jello, it doesn't work very well. And we end up with things like Alzheimer's, Pick's disease, Huntington's disease. Things can be related to HDAC6, although they're also related to other systems that are malfunctioning. It's related into cancers, kidney disease, some of the cardiovascular disease for the same reason. If you can't transport the proteins, they just get thrown out and they don't get lysed like they should. They get built up too much and they're put on the surface, and there's all sorts of various things that can happen. So this is a classic case where you've got one gene, and as far as syndromes are concerned, as morphological outcomes from it, you would think of it as a pleiotropic effect, right? It's one gene having all sorts of potential effects. Now, a lot of those effects are good. It's doing its job throughout the cell, but when it's mutated, it can end up having less than desirable outcomes. All right, so let's try a couple other little pathways fairly quickly. One is mitochondrial fate. I just said that. I said, okay, you've got these mitochondria. They become dysfunctional for various reasons. They become, there could be oxidative damage. They can build up too much of their own internal ROS. They can become depolarized, and they can, they can lose their system. There's all sorts of things that can happen to mitochondria to make where an individual mitochondria is not functional the way it should be, right? So when that happens, you need to get rid of them. You need to break them down. They're an organelle that we want to remove. And that process, instead of calling it autophagy, is called mitophagy, okay? Mitophagy is the way that most of us would pronounce it, autophagy, mitophagy, okay? So it's the way to form. So mitophagy. You see it, British pronounce it mitophagy, but anyway, doesn't matter. This is another system. The greens here are all they're all proteins that are involved in this process. One of the important ones are these ubiquination proteins that ubiquinate structures, okay? So they ubiquinate the mitochondria just like the strings, just like they do some of the other things. Parkin is one of the important ones, and there's a number of diseases that are due to mutations in the Parkin gene, as well as the PINK1 gene that's over here. These are all, sort of causing or detecting variation that's occurred in a bad way. And it can be driven by lots of things. Again, ER stress, because mitochondria is important there, depolarization, hypoxia, that's all sorts of things that can drive it. The point is that it takes, again, a lot of proteins are involved. A lot of genes are involved, a lot of interrelated gene groups that are involved, and you can have pretty much a change in any one of these places will have some effect. How do we know that? Because we go in and we knock stuff out and we look for it. We'll talk about that later with animal models, but when we want to know HDAC6, we have variants of HDAC6 where we have deleted particular sections. I mean, that's the way that we figured out that they actually had domains earlier. And the way you figure out that you have a domain there is you go in and you take the actual message, or you take the DNA itself, you make a deletion called a delta, and you delete a section, and you delete a known section, and you see what's not working. That's the way we would do it. So the way we figure out these domains is by these deletions. And it's the same thing when you talk about any of these. We come out and we just stop the whole protein, right? There are ways to do that in cells, in cell lines. There's ways to do it in whole animals. We have whole animal mice that are P62, not H.6, but P62 deficient. We've knocked it out, and we watch what happens to the whole organism, as well as knockout cells. We have H.6 knockout mice where we have downgraded those and can look at the outcomes, right? Just a couple more. One cell fate. Okay, whole cell. Now we're not talking about just the proteins itself, but we not only do we get rid of mitochondria, but we have times to get rid of cells, and it's called apoptosis. And apoptosis is programmed cell death. Program being that there's a process that goes on. Okay, some cells go through this regularly, right? When cells mature, they have a certain lifespan, and then they degrade and disappear and replaced by other cells. Skin cells being a good example of that. Other cells is caused by external problems, external inputs, radiation, genotoxic drugs can cause it, heat shock and nitric oxide, NO levels being high, hypoxia, damage to the DNA, all sorts of things. So just like you can have a misfolding protein and stuff, you can have lots of things that can damage the nuclear DNA. And in response, the system says, okay, these cells need to be turned off, right? And so there's a number of pathways to that. What's different here is that unlike the last pathways where there are lots of convoluted interrelations early on, the apoptosis system has a number of triggers, but they all trigger down to P53, a protein that once it's phosphorylated, moves into the nuclear region and hits a number of DNA spots. And depending on where it goes, and depending on the severity of the system and the way that it's phosphorylated, that signaling in P53 can turn on different outcomes. Okay, some of the outcomes are simply like things like cell cycle arrest and cell senescence. Then you've got the full apoptosis pathway where you take the whole cell, the cell's degraded by, again, coming out, making, triggering on all this suite of proteins here, working through the mitochondria to actually then have the whole cell just basically degrade. And as you see over here, this nuclear collapse and dissociation. There can be other things that go on. You can simply have, it can trigger repair and say, okay, there's some things going on in DNA, but I can repair it. There can be some feedbacks and other things that go on. But the point is you get this input, you got a fairly short pathway response, and then you get a direct input into the DNA structure of the system that will lead to apoptosis. And then, one that we probably don't think about too much, but I think is real important is her circadian rhythm. If you ever go out to, lucky enough to go out to a bat cave, watch bats come out, or maybe Carlsbad, if you're ever out that part of Mexico, bats, complete darkness, deep in a cave, all of a sudden at a certain time of night, here they all, millions of them fly out, okay? Then in certain times of the morning, millions of them fly back in. It's pretty cool. Watching them come out is pretty cool. Watching them come in is even cooler because sometimes they're flying at 2,000, 3,000, 4,000 feet and they come over the cave and they just stop and they just fall. And you hear them just going shoop, shoop, and it's pretty impressive. But how do they do that? Well, they do it through a circadian rhythm. They have a natural inbuilt rhythm system that works. And humans and mammals have the same thing. If we look at our system, we sort of say, okay, if we wake up at 6 o'clock in the morning as a wake up time or whatever, and that we become active, we have a rise in blood pressure, melatonin is stops secretion. We often find that we need to go to the bathroom that time. Alertness peaks and continues on around noon. We get some of our early afternoon, 2:30 or 3:30 or so, depending on who you are. You start to see some of your physical strengths increase. And then as we start to shut down, 7 p.m. or so, 9 p.m., we start having melanin production, you get a little sleepy. And then as you go to sleep, then you suppress various things. Body temperature tends to drop a little bit. Then we cycle back through this whole process, okay? Some of us, have irregular parts of it, and certainly in college, we tend to be up past midnight a lot of times, and we don't get the sleep we need, etc. If any of you have ever worked the midnight, the graveyard shift, it's, for some of us can work it quite fine. Others like me, I just hated it. So I would, we work in the lab really late. It's one thing, where you work all day and then you work into the night. But when you had a regular shift pattern where you had to go in at midnight and get off at say 8 in the morning, I was just all messed up. I certainly wasn't alert. I tried to sleep during the day and it just never worked out well. Others seem to handle it quite well. They can change your rhythm. But anyway, you have a circadian rhythm. Well, all of these things that are happening, okay, you're having things like melatonin secretion stops. You're down-regulating the genes that produce melatonin, their process. If testosterone goes up, okay, if you're male, well, you're upregulating the genes that are involved in testosterone. Blood pressure changes, body temperature changes. Those are controlled by genes upregulating or downregulating the system, okay? Now, where it primarily comes from, at least in mammals, is through the hippocampus. So you have the ocular nerve, okay, the optic nerve here coming in. Right at the base of that, where the nerve comes in, you have the chiasmatic nuclei. There's a series of cellular structures here that are receptive of that light, okay? And they're found, and they actually have one of the regions where you secrete certain hormones from that region out to other cells, trigger certain things that go on in the brain, okay? And so you have this extracellular signaling that comes in. There's calcium changes along the membrane. There's other things that go on. And in doing so, that starts off this MAP kinase signaling pathway, or CREB pathway. And CREB is actually found on chromosome 4, right here on the long arm, down from the centromere. And you start having production of that. You upregulate that particular protein. It gets phosphorylated. Once it's phosphorylated, it's able to move back into the nucleus. And when it does, it hits up a number of DNA sites, okay? Different regions, different chromosomal regions, and those have different outcomes. They start producing products that are transcribed. They go out and get translated. You get feedbacks from those back into the system. Other DNA is activated, etc. Ultimately, from all this, you get DNA activation for a whole pathway that then sends out this clock-like output, right? So you get a light input. Cells respond to that light input. You get genes turned on. You get genes phosphorylated so that they can do their activity. That's one of the ways that proteins function, right? Genes get phosphorylated, but proteins get phosphorylated. So they make their tertiary quaternary structure. They get phosphorylated. That causes them to be active, right? In this case, their activity is to lead a pathway that's ultimately going to come in and link onto some DNA, trigger the transcriptional units to become active, the enhancers, and you end up with products being produced. So that's one of the points. Now, finally, the last one I want to talk about are things like addiction and pathways. And these are interesting because we see a slightly different response, right? Let's think about cocaine or alcohol. They work somewhat similar ways. When you have neural cells, you have this neural synapse, and what triggers in between those synapses, okay, are dopamine molecules. And dopamine is a fairly small hormonal structure. It looks like this. They're released from one side through energetic system. There are receptors found on the others, okay? That's where you see it going over here. And they're collected, and that causes stimulation. Okay? It causes this one, so that's the signaling pathway that occurs. In a normal system, and your brain's working like you're sitting here now, your brain's working. There's dopamine going back and forth and degraded. There's feedback loops and all sorts of things that go on. When you introduce drugs, some other drugs like cocaine in particular, you have an overstimulation of this structure, and then the dopamine they just release, massive release. You get massive stimulation. You get a high from it for a period of time, etc. And notice that here we go through this kinase, this PKA process. Okay, Well, when you have this cocaine, other or high stimulation of neural systems, cocaine being one of the things that'll do that, then it definitely pushes this system even further. Recognize this protein right here involved again is one that becomes phosphorylated in the last discussion and enters DNA. And what normally happens is that you will get a reduced drug award signaling. You'll get these things that sort of while you'll get locomotor response and you'll get triggering, particularly if you go into the sympathetic nerve system, you'll get triggering of locomotor responses, be able to, oh, I can dance really well or something, whatever. But you also get a reduced drug reward process, and eventually this loops back. And so while you get a burst of activity at first, then you slowly, because your receptors, you lose out of it. That's from just, a normal introduction. When you have acute or long-term exposure, you don't get the same pathway, okay? What happens in this pathway is that both in the systems that are firing, you don't get the same amount of these dopamine hormonal responses coming forward. There's not near as much of them because the protein that's involved in pulling those back in and degrading them is more active. Okay, there's blockers. The receptors, there's fewer receptors that are going to respond, and so you don't get the full response. Now, ultimately, you will get the full response. Okay. But the DNA that's turned on there, the genes that are turned on, notice this is a different pathway. This is a FOSB pathway, and you end up with an outcome that pushes back. So long-term exposure to it, you don't get the high as you would, right? You need more and more and more and more to move you from this level of excitation back up to the level that you first experienced. But in doing that, this becomes an addictive problem, right? The addiction part of it is that you get this response here, and your body is sort of, there's some secondary outcomes of these exposure where you start to crave it. Okay? It looks like with alcohol, you get a similar type of issue. Normal systems firing along, you got the receptors. When you reduce alcohol, there's a whole series of receptors that respond. And those receptors fire in through another activation system, and they fire in and increase DNA function over here, right? Once that DNA is activated, then you get normally you should get a feedback loop. Okay, so at some point you get this, from alcohol you get a short high for it to last any length of time. This feels good, everything's great, and then there's the feedback. Slowly you start going, yeah, well, it's enough of that, and that's a feedback loop where you actually end up with proteins being removed outside of the cell. The ethanol is there, and then you get a return back in with some phosphorylation and all sorts of things back to DNA. Ultimately, then you should get a decrease in intake. You should activate the cells itself are not pulling in the ethanol at the levels that they were. Okay? That's the normal process. It also turns out that alcohol has an epigenetic effect itself. Ethanol can enter the cell, the cytoplasm itself, ultimately getting into the nuclear region, and can cause all sorts of modifications of the histones themselves. And activation, so it can turn on and turn off genes in certain ways by its epigenetic modifications. We'll talk about those a little bit later. So not only does it directly affect DNA through a pathway connection that pushes, but the ethanol itself is truly directly impacted, in that it physically, as a poison, can cause epigenetic modifications. Okay? Now, what happens when you have chronic things, you have ethanol in the system constantly, is once again you have a decrease in the production of these dopamine release. There's fewer receptors that are responding, and so you eventually get this inhibition, resistance to inhibition. Okay, so what that means is there are natural systems that went through calcium activation that kind of stop the ethanol transport, right? Because the ethanol is going to move in. So they kind of block it and say, okay, there's a lot of ethanol in the system, but we're not. if chronically, you eventually get to where there's modification of these systems where there's much more ethanol allowed into the system, and there's much more response, at least, to the ethanol. And so your resistance goes down, okay? And when that resistance goes down, more signaling occurs, but this time it's different signaling. It's through the MAP kinase pathway, Merck kinase pathway, and you get a lot more input into the system, okay? And you end up with sort of a reverse, unlike this, which is pathway works to decrease it. Through this pathway, again, the PHOS-B pathway, which we saw similarly in cocaine, you get there's a positive response outcome, and so it leads to more and more and more. So we constantly want more and more and more and more of this through this pathway. So what am I doing here? Well, again, I don't want you to memorize any of this. Okay, look it up. You can always look, pull back your notes if you need to see it. I want to point out there's pathways require lots of genes, because each of these green things is a gene that's been activated at certain times. There's upregulation and downregulation of genes in certain ways, depending on the amount of things that come in, how they're coming in, the timing, and they become important. So, just having a drink of alcohol, you think, well, I had a drink of alcohol, it goes into my liver, and I kind of break it down, everything's good. Yeah, except that it's also traveling around to the brain, to the blood, crossing the blood barrier to the brain, causing other things to go on. And you get these, you're activating a large suite of genes to do certain things, right? And they're being activated for reasons that they aren't naturally activated to do. So it's kind of an interesting problem. And we'll talk more about that. We talk about behavior and some other things, okay? And the final thing I wanted to show you was that there's this KEGG pathway page is a really good pathway page. And I'm going to ask you to look at it in one of your exercises. But if you're really interested in sort of this, we can, if I can find it here. Yeah, there we go. If you go to the page, if you click on that or go to that page, you'll go to this pathway database. And it has all sorts of pathways that are involved in information processing, genetic information processing, whatever. And if you click on it, it'll tell you the pathway. It'll show you, is where I got most of those pathways. It'll actually show you a nice little diagram of what the replication pathway looks like, what genes are involved, how they work and function. And it's a nice little option for all of us. Whether you're in the medical field or whatever you're doing, you're interested in how a system works. This is a nice little page that you can look at. Okay, so we'll finish up with that. All right. Well, that concludes what we want to do. Just a simple cellular review. We'll now move into things that are more directly related to our human genetics components and talk about that. Those will be on the next lectures.